It's 50 years this year since President Nixon signed the National Cancer Act. 50 years. And I know that so few of you were around then, because you were just not even twinkles in your mummy's eye. But nevertheless, some of us do remember the euphoria at, oh, cancer, make cancer history, basically, was the message. Fantastic. So how have we done? It's set to displace heart disease as the #1 killer in the Western world. Not good. And 1 in 2 people in the UK born after 1960 will be diagnosed with some form of cancer in their lifetime. Those born before 1960 have a little bit more leeway, but not very much. And a few scientists have admitted that such improvements as there have been is largely due to actually people giving up smoking, which they have done in the Western world in huge numbers. So we can be forgiven for asking, what have scientists been doing all this time? Well, in the mid-1970s, they believed that the answer was that cancer was caused by mutations to oncogenes, and all we had to do was design some very expensive gene-targeted therapies, and we could treat it. So they looked for these oncogenes, and they found a random collection of mutations with no one mutation or any combination of mutations being responsible for initiating the cancer. The mutations for each cancer type differed from person to person, and sometimes they even differed from cell to cell within the same tumor. The American Cancer Society admitted that cancer appeared to be a collection of more than 100 different diseases. You can see why. And a study attempting to find the mutations driving metastasis, which after all is what kills us, found not a single common mutation. So where's all the research funding gone then? Well, the US National Institutes of Health have spent over $1 trillion on this, but the therapies have been a great disappointment, and except for a couple of rare cancers, they barely improved the death rates from the 1950s. And the American Cancer Society has now admitted that genes contribute no more than 5% of our cancer risk. So those genetic mutations turn out to be due to modifiable epigenetic factors. This is what you treat every day of the week. Yet 95% of the research funding is allocated to genetic research, and prevention, which is targeting the modifiable epigenetic factors, accounts for 5%. I'm sure I'm not alone in thinking that they have really got this the wrong way around. But I hear you say, what about those oncogenes? And what about BRCA1 and breast cancer? Well, let's have a look at BRCA1. 50% of women who carry the mutation never get breast cancer or any kind of cancer. And in women with early onset breast cancer, there was no significant difference in overall survival between patients with either the BRCA1 or the BRCA2 mutation and those without. And when you look at the purpose of the BRCA1 gene, it shows that it has many roles in the body, but one of which is repair of DNA damage. So don't you think that actually that might be the cause of the cancer, failure to repair DNA damage, rather than there being an oncogene itself which causes cancer? Cancer is also not a disease of aging, not any longer. Rates of cancer incidence are increasing in children and young people, and in the US, after accidents, cancer is the next most frequent cause of death in children. That's pretty shocking. So scientists have slightly moved on, because we discovered 2 years ago that they are now looking for clues to how cancer is born. The idea is that scientists have teamed up to search for the earliest signs of cancer so that they can squash it before birth, basically. But even they admit that this is a pretty hopeless quest. So to my mind, this is a fairly clear admission that the genomic approach has not worked. But the how cancer is born approach is also doomed to failure, in my opinion. Why? Because every cancer cell in our body has the capacity, or every cell in our body has the capacity to become a cancer cell every moment of every day under the right conditions. And in fact, those of us without a cancer diagnosis all have 75 million cancer cells in our body right now. So what's keeping them in check? Well, our immune system. Immune system surveillance and targeting is defective where cancer develops. So wouldn't these scientists really be better off looking at the immune system? So that's exactly what we're going to do. So the immune system is the most important defense against cancer growth. Immune cells of the innate immune system are continually surveying the body for cancer cells. And when they find them, they trigger cytotoxic lymphocytes and natural killer cells to go out and kill them. Great. And in turn, these dying cancer cells release damage-associated molecular patterns. These are recognized through pattern recognition receptors, which themselves can kill tumor cells, but they also trigger a response from the adaptive immune system, which sends out cytotoxic CD8 T lymphocytes. Brilliant. And this is pretty much always sufficient in a healthy immune system. So surely we should be actively supporting our immune system all the time. And I know you all know how to do that because we've all been doing it for COVID. So, and another word about antioxidants. I promise I'm not really against antioxidants, but the immune system works by generating free radicals to trigger apoptosis in the cancer cells. So the last thing we want to be doing is giving antioxidants to neutralize them. There we are. I won't mention it again. So we've done the immune system. We've discovered that the genomic approach is wrong. The how cancer is born research will never work. So what should we be looking at? Oh yes, the mitochondria. So Sarah has already mentioned Otto Warburg. Was you, Sarah, wasn't it? Yes, that's it. So back in the 1920s, Otto Warburg, who got a Nobel Prize for this, by the way, he found that cancer cells operated aerobic glycolysis. Now, we talked about anaerobic glycolysis, which is where you've run out of oxygen, basically. So you're having to operate anaerobic glycolysis in the cytosol. But this isn't that, because you haven't run out of oxygen. This is the cell choosing to operate glycolysis in the cytosol, despite the presence of oxygen. And he hypothesized that this was due to impaired mitochondrial respiratory capacity in the cancer cells. And he said that there's only one prime cause of cancer, and that's the replacement of the respiration of oxygen in normal body cells by the fermentation of sugar. And this became known as the Warburg effect. So naturally, with a new and innovative idea, he was ridiculed, he was reviled, he was knocked off Facebook, and well, we didn't have Facebook then, but you know, you get the general picture. But just when the tide should have been turning, gene theory arrived. So that was the end of that. But all was not lost, because then who arrived but Professor Thomas Seyfried? And he wrote this great book, Cancer as a Metabolic Disease. I say it's a great book. I confess I haven't read it, but that's because it's hugely, well, it's huge and hugely expensive. But I have read his studies. And actually, if you want another book that talks about his approach and what else is going on in cancer research at the time, I thoroughly recommend Travis Christofferson's Tripping Over the Truth. Very good. So let's see what Seyfried had to say. Well, he confirmed that nearly all cancer cells used energy derived from aerobic glycolysis. So well done, Warburg. But not only do they ferment glucose in the cytosol, they also ferment glutamine in the TCA cycle. Ooh, that's new. And Seyfried diverges from Warburg in finding that cancer cells can have intact mitochondrial metabolism with the ability to produce ATP through OXPHOS. So it's like they're producing ATP from OXPHOS, and then, oh yes, they're going into aerobic glycolysis as well, with a little bit of extra ATP. So aerobic glycolysis has 3 advantages he found for cancer cells. Firstly, although it only produces 2 molecules of ATP compared to the more than 30 from OXPHOS, nevertheless, aerobic glycolysis happens so much faster, and in cancer cells, it happens around 100 times faster, that you end up with a net effect of greater ATP production. So that's quite interesting. And then you get the lactate, of course. Now, in a cancer cell, lactate is a fuel. So you're generating ATP through aerobic glycolysis. You're getting lactate as a byproduct. You can then recycle it as your next fuel. This is not good. And evading OXPHOS means that you get fewer free radicals produced. And with fewer free radicals, there is less to trigger apoptosis in these cancer cells. So you can see there are seriously good advantages for a cancer cell. And metastasis, he found, is associated with enhanced mitochondrial respiration and biogenesis. And inhibition of OXPHOS actually suppressed metastasis. So that was all quite interesting. So is there any independent corroborative evidence for this? Well, there is increasing evidence that cancer has a metabolic origin. We can see upregulation of glucose transporters and glycolytic enzymes in tumor cells. And here's a 2016 meta-analysis, which looked at more than 200 studies and concluded that the most important difference between normal cells and cancer cells is in how they generate energy. So yes, independent science is getting there, slowly, but it's getting there. And then, of course, there is a large connection between metabolic disease and cancer. So we can see this because we see all the time obesity and diabetes associated with incidence of many types of cancer. And insulin and IGF-1 can trigger cancer initiation. Cancer cells consume blood glucose 50 times faster than normal cells, and breast cancer cells have 3 times as many insulin receptors. And here's a meta-analysis of 19 studies found that serum glucose was associated with incidence of both hormonal cancers and those driven by IGF-1, and an NHANES study, that's the large US population study they run every 8 years or so. This found that blood markers of insulin and glucose metabolism were predictive of cancer mortality. So the science is definitely getting there to support Warburg and Seyfried's approach. Now, Seyfried did 2 other things as well, which I think is really cool. So he disproved the genomic theory as follows. He took a tumor cell, he took out the tumor nucleus containing the mutated oncogenes, so-called. He put it into a healthy cell without a nucleus, and nothing happened. It remained a healthy cell. Brilliant. So that disproves the genetic theory, according to him. So then he went on to show something even better. So he took a healthy cell, took a healthy mitochondrion out of it, put the healthy mitochondrion in the breast cancer tumor cell, and it became a healthy cell. Fantastic. Isn't that great? So I'm going to come back to Thomas Seyfried because I'm not done with him yet, but a few things we need to bear in mind when we're dealing with cancer patients. So above all, we don't want tumor cells to be healthier. We actually want them to commit suicide. We also don't want to improve their mitochondrial metabolism because that's not going to help at all. We also have to remember that cancer is extremely flexible and it can evade just about everything that you throw at it. So while an obvious strategy is to eat 0 carbohydrate to reduce that glucose, and that's very, very sensible, nevertheless, the cancer cell's need for glucose is such that it's going to initiate gluconeogenesis, which is what Sarah was talking about, by breaking down proteins. And this indeed is what cachexia is. And you don't want to lose your structural proteins because you're not going to get them back in a hurry. So obviously also supporting the immune system is a priority. So let's look at our mitochondrial therapies for cancer. So caloric restriction and fasting, yes, great. Ketogenic diet, Seyfried did one of these studies, terrific. And extreme temperature therapy also proved to be quite good. So this was hyperthermia, not hypothermia. So I can't honestly recommend any more because there aren't the studies for it. But I thought we would just take a look at the American Cancer Society's dietary recommendations for cancer patients. Yes, quite. So back to Thomas Seyfried then. So he says that whereas the genomic theory treats cancer as more than 100 different diseases, as we saw, he treats it as just one disease, as indeed Warburg did. So using that logic, he came up with just two forms of treatment. First one is known as the press pulse cancer treatment. So this is restricting glucose and glutamine, and you can see the logic for this. So you do that in a cyclical fashion. But reducing a glutamine is really difficult because it's one of the most abundant amino acids. What on earth are you going to do? But the good news is that cancer cells cannot use ketones as fuel. They may be able to use lactic acid, but they can't use ketones. And then his next treatment was the restricted calorie ketogenic diet, the RKD. So this proved to be extremely successful because it was anti-angiogenic. So the last thing you want in a tumor is new blood vessels growing because that's just going to bring more nutrients in. It was pro-apoptotic, fantastic, they're all committing suicide, and it's anti-metastatic. Well, that's great. And then he teamed up with Dominic D'Agostino, who did hyperbaric oxygen therapy. So this triggers a lot of reactive oxygen species, which induce apoptosis in the cancer cell. So they did a trial, and the RKD therapy increased mean survival by 57%. When you added in the hyperbaric oxygen therapy, mean survival increased to 78%. So that's pretty good. So their therapy then strongly influenced this place. And I know a number of doctors who have successfully sent patients there, although it doesn't work for everyone, but what does? So this employs Seyfried's work and 4 of our mitochondrial therapies: fasting, ketogenic diet, hypothermia, and hyperbaric oxygen, combined with very low dose chemotherapy. And admittedly, they've only written their own studies, but nevertheless, they've been published and they seem to be very successful at it. Again, there's no independent corroborative evidence because who's going to get funding for a study like that? So let's look at our mitochondrial remedies for cancer then. And we've actually got quite a lot. So I've got a few things to say about that. First of all, most of these, and particularly the bioflavonoids, they manage to damage cancer cells, but leave other cells undamaged. So this is in stark contrast to chemotherapy and radiotherapy, which damages everything in sight. So some of these have been used in studies to enhance the effects of chemo and radiotherapy in so-called resistant tumors. Now, how this works is the patient's done chemo and radiotherapy and it's not killed the cancer. So then they've added in these bioflavonoids and it works. So the scientists have explained this by saying, oh, the tumor became sensitized by the bioflavonoids so that the radiotherapy and the chemotherapy could work. But my view is, maybe they didn't. Maybe the chemo and radiotherapy was never going to work and the bioflavonoids did all the work. I don't know. There haven't been enough studies to say. But another interesting point is that virtually all of those remedies reduce the symptoms of chemo and radiotherapy. So if your patient is insisting upon having conventional treatment, it's worth supplementing these as well. There is no mitochondrial remedy that is specific to any one type of cancer, as Warburg and Seyfried would have predicted. But I do have a caveat, and that's that some of them in excess are carcinogenic. So just be very careful of these. And here are my top 4 remedies. No great surprises there, probably. And then a year ago, we had this headline: government may intervene to curb cancer cure propaganda. So what's all this about? Well, this is ministers thinking of expanding the Cancer Act to police medically unproven procedures. That's everything I've just been talking about, which is proven actually, but not to their satisfaction, and bring in tighter regulations to protect people from fake cancer treatments and dodgy practitioners. Well, dodgy practitioners, sorry to say, is you lot. So keep your eye out for this, but I foresee trouble ahead. Right.
Thank you, Rachel. I'm sure we'll have lots of questions. I have one here, going back to the beginning of your lecture. How would you recommend we use antioxidants then? Periods with and without, or only when ill?
Oh dear. This antioxidants issue has really got you going, hasn't it? What can I say? I mean, antioxidants have a place. For instance, a lot of the things that, well, I mean, take Sarah's talk with her high dose bowel tolerance vitamin C. Vitamin C is an antioxidant, but that's not the only thing it does. It's doing what Sarah wants it to do, which is wiping out the microbes. So in that case, it's highly appropriate. What was the question again? Sorry.
I don't think you're going to have a full answer to it. The question was, how would you recommend we use antioxidants then? Periods with and without, or only when ill?
Well, not when ill with cancer, that's for sure.
Optimizing the diet.
Yeah. Don't use antioxidants in cancer patients, I would suggest, because the immune system is trying very hard to kill the cancer cells, and you're just going to neutralize it by giving antioxidants at that point. I mean, they have a role in the body, but I am trying to focus on the mitochondria today.
Yeah.
And I would say that for the mitochondria, they don't really have a role, simply because, as I said before, you'll never get them into the mitochondria. What you can do instead is upregulate the endogenous antioxidant in the mitochondria.
Thank you. Let's take some questions from the floor.
What about caveat include vitamin C?
Which caveat's that?
Antioxidants with cancer. Would you include vitamin C in that?
Right.
Or would you use vitamin C?
I think you would have to be very careful. And again, I have to stress, I'm not a practitioner. So if you think the cancer has arisen because you've got microbes in the gut, maybe vitamin C would be highly appropriate to get rid of those, and you just accept that it's going to neutralize reactive oxygen species. What do you think, Sarah? You're going to disagree with me, aren't you?
I don't know.
Well,
but my guess
no one does.
Local effects, systemic effects.
Right.
And what we do know about the way in which immune cells kill cancer is a very local effect.
Yes.
And I see antioxidants working as we have what I call, like again, Quidditch, coming back to Harry Potter. We have, frontline seekers or whatever, grabbing the spare electrons, and that would be things like glucose can say 10 times they're in the front line, and they're in their timing now. And then they pass them back to second line antioxidants like vitamin D, vitamin C, vitamin K, vitamin all that. And then they pass them back to the ultimate repository. That's where you have vitamin C gram. So I would say vitamin C is essential. There's a lot of cancer. And the point is, biochemically, vitamin C looks exactly like sugar. In fact, all other mammals can make their own vitamin C, and they make it from sugar, and massively upgrading your production in response to infection. Humans can't do that. So that's why we need to supply the extra so they can take what's needed.
Right.
Yeah.
Where he looked at vitamin C. He was actually doing work with schizophrenic patients, and some of them had cancer alongside, and he was giving B3, which is obviously very relevant for today, and vitamin C. And he showed that by giving 10 grams of vitamin C orally and B3, that people at any stage of cancer, but particularly stage 4, were able to live 9 months longer just on that regime alone.
Fantastic. Thank you, Andy.